I'm Adam Sperling. I'm one of the members of the Dana-Farber Multiple Myeloma Group, and it's my pleasure today to give an update on CAR T cells and multiple myeloma. So these are my disclosures. I've done a little bit of consulting for both Adaptive and Roche, although neither of these have any relevance to the talk I'm giving today. This is an outline of the talk, so I'm going to start with an introduction on cell therapies and multiple myeloma. I'm going to talk a little bit about CAR T cells and the primary target, which is the B-cell maturation antigen. Then I'll talk a little bit about other targets in multiple myeloma and finally finish with next steps and future directions. So we've known for a long time that the immune system is often abnormal in patients with multiple myeloma, and there have been a number of efforts throughout the years to target the immune system to try to improve therapy for multiple myeloma. And this goes back to drugs like thalidomide analogs, thalidomide and lenalidomide, which certainly alter NK and T cell function, monoclonal antibodies like daratumumab and elotuzumab that target the immune system or alter the immune response to multiple myeloma, attempts to develop tumor vaccines, which are still ongoing, and then of course more targeted approaches where we either re-engineer or retarget T cells to attack multiple myeloma cells, and that's really what we're going to focus on today. So there's really sort of two major approaches to targeting T cells to multiple myeloma. The first on the right is chimeric antigen receptor T cells, and that's where we re-engineer the T cells using genetic approaches to express this special chimeric antigen receptor. This chimeric antigen receptor can then bind to BCMA or other targets on the surface of multiple myeloma cells. It brings the T cell close and in the process activates the T cell, causing it to target and kill the multiple myeloma cells. There are other ways of targeting T cells to tumor cells, and one of them is these bispecific T cell engagers. So these are molecules that have two ends, and this is demonstrated on the left. One end of the molecule binds to BCMA, and the other end binds to the T cell receptor on normal T cells. In doing that, again, it brings the T cells close to the multiple myeloma cells and activates them, which allows them to kill the cells. So these are all examples of what we call adoptive cellular therapies. But what that means is where we take cells, or CAR T cells are, at least we take cells out of a patient. So these are the T cells we take out. We do this through apheresis, which is similar to the process that we use to collect stem cells in patients who are undergoing autologous transplant. The T cells are removed, and then they are engineered in the lab. And the approach we use for engineering is primarily done through viral transduction, although there are other approaches. So this takes DNA that we've made and inserts it into the T cell genome, which causes it to express these chimeric antigen receptors. Once the engineering is complete, and there are various processes, but this takes anywhere from 2 days to 2 weeks to do, the cells can then be infused back into the patient after they've been given what we call lymphodepleting chemotherapy. And the lymphodepleting chemotherapy is given primarily to decrease or tamp down the patient's own immune system so they don't reject the cells, and so there's room for these cells to grow, develop, and target the tumor. Once they've been infused, we generally just monitor patients and don't provide any additional therapy, although there are questions about whether or not that's the best approach. So there are a number of different targets in multiple myeloma, many of which have been tried or are in the process of being tried. And some of these are sort of canonical proteins that are expressed on plasma cells like CD38 and CD138. Other ones like CD19 are expressed on mature B cells. Sometimes are expressed on multiple myeloma, but these have really been used primarily for other types of blood cancers like lymphoma or acute lymphoblastic leukemia. And then targets like SLAMF7, which is the target for elotuzumab. People are attempting to develop CAR T cells, but probably the two most successful targets and the ones that I'm going to talk about today are the B-cell maturation antigen and this protein GPRC5D. So what is B-cell maturation antigen, or BCMA? So BCMA is a member of a family of receptors called the TNF receptor superfamily. And these receptors are expressed almost universally in multiple myeloma cells, so it's very rare to have multiple myeloma cells that don't express it. And their expression is largely restricted to normal plasma cells, multiple myeloma cells, and some mature B cells. So it's not really expressed on many other cells in the body. And so that's very important because it means you can target it without targeting other cells that might cause toxicity. And the other thing that we think is important is that we think BCMA is actually important for multiple myeloma cell and plasma cell growth. And so we think it's difficult for the plasma cells to lose BCMA completely because they do need some BCMA activity in order to survive. And that also makes it a good target because it means it's not easy for the cells to lose. So there have been a couple of different products that have been used to target BCMA. And the first that came along was ide-cel, or BB2121, which is an anti-BCMA CAR T cell. So this is what we call an autologous CAR T cell. So it takes the patient's own T cells, we remove them, and transduce them like I discussed earlier. It has a few features that are can vary between CAR T cells. So it uses this 4-1BB and CD3 co-stimulatory and intracellular signaling domains, and these are important for how it signals to the T cell and induces the T cell to kill multiple myeloma. But actually, most of the CAR T cells in multiple myeloma have used this structure, and it's been tested primarily in patients with relapsed or refractory multiple myeloma. So this was done in the KarMMa study, which tested ide-cel, or BB2121, as it was called at the time, at 3 different doses in patients with relapsed and refractory multiple myeloma. And so you can see here in the outline of how the study was done is that patients were identified, they then had their T cells removed, and then the T cells were manufactured. Now the manufacturing itself takes about 10 to 14 days, but there's additional testing that has to be done to determine the quality of the transduction, make sure that there's no contamination, things like that. So the actual manufacturing takes considerably longer. Patients then received the lymphodepleting chemotherapy. This is a combination of fludarabine and cyclophosphamide. They received 3 days of it, followed by infusion of the BB2121, at which point they were monitored. And this trial included patients who had received at least 3 prior lines of therapy and had been exposed to an IMiD like lenalidomide, an antibody like daratumumab, and a proteasome inhibitor like bortezomib. So in this trial, the response rates were very high. So the majority of patients responded overall. About 75% of patients responded, and many patients had very deep responses with a complete remission or a stringent complete remission to response. And you can see that the response rates were a little bit higher at the higher cell doses. And so that 450 million cell dose has been the target dose going forward. Now the duration response is a little bit longer with the higher doses, but I think the key thing here is that the vast majority of patients still progress, and it doesn't take that long. So it's about a year, 10 months to a year. Most patients do progress. So this is a very effective treatment. It induces very deep responses, but unfortunately patients still do progress. So this led to the FDA approval of ide-cel. It was approved at a dose of anywhere from 150 to 450 million cells. So the 450 million cell dose is really the target dose, but in some patients we're not able to manufacture that many cells. And so it does allow patients to be treated even with lower cell doses because it does seem to be effective even at those lower cell doses, even if it's slightly less effective. It was approved for patients with relapsed and refractory myeloma. They have to have had at least 4 or more lines of therapy and must have been exposed to all 3 of our standard classes of drugs. Now, there are a number of ongoing studies that are testing ide-cel in additional settings. So the KarMMa-2 study is testing it both in that same relapse refractory setting, so it's generating additional data, as well as in earlier phases, so patients who relapse early after transplant or after induction, to see if maybe it will work better in earlier lines of therapy. The KarMMa-3 study is a large randomized phase 3 study that's been required by the FDA as part of their approval process, and the early word is that this is a positive study, although not all of the details have been presented yet. And then there are other studies, again, looking at ide-cel, and this is not a comprehensive list, but looking at ide-cel either in combination with other agents or again in earlier lines of therapy. And so I think over the next couple of years, we'll have more information and have more data on how to use ide-cel and whether it works better in earlier lines. Now, there's a second CAR T cell that came along a little bit after ide-cel called cilta-cel. And this is very similar. It was actually originally developed by a company called Legend and developed in China, but was then has been marketed in collaboration with Janssen here in the US. It's also an autologous CAR T cell. It has the same 4-1BB and CD3 zeta signaling domains. The one difference is the way that it binds to BCMA. It binds with 2 BCMA targeting sites, and so may bind a little bit tighter, although it's unclear exactly what the role for that is in its activity. The design for the CARTITUDE-1 study, which was testing cilta-cel, was very similar to the KarMMa study. So it tested some different dose ranges, but the median dose was about 0.75 million cells per kilogram. And the structure in terms of the apheresis, manufacturing, and lymphodepleting chemotherapy was all very much the same as in the KarMMa study. Now, the response rates have been incredibly high, actually higher in CARTITUDE. So 97% of patients responded to the treatment with cilta-cel, and the duration of the responses has also been longer. So this is the progression curve, and you can see that even out as we get out towards 2 years, we haven't reached even half of patients progressing. So this is significantly longer than what was seen with ide-cel. This data led to the approval for cilta-cel. Again, it was approved at this 0.75 times 10 to the 6 cells per kilogram. And otherwise, the approval is very similar to ide-cel, 4 more lines, and had to be exposed to all of our standard agents. It also is being tested in additional studies. So CARTITUDE-2 is again testing it in relapse refractory setting, as well as in combinations with other myeloma agents. And then there are arms that are going to be testing it in earlier lines of therapy. And then they also have a randomized phase 3 study that is ongoing. So I thought I would just compare some of the different anti-BCMA therapies. So here, this is ide-cel, cilta-cel, so the 2 CAR T cells. I've also then put up information on teclistamab, which is one of the bispecifics that was just approved, and then belantamab mafodotin, which is an antibody that targets BCMA. So these are really the 4 approved agents that target BCMA in multiple myeloma. And you can see that there are some differences in terms of their efficacy. So for one, the response rate seems to be the highest with cilta-cel, 98% response rate in the clinical trial. That's a little bit higher than ide-cel, which was closer to 75%, and higher than either teclistamab or belantamab, which were lower. The complete remission rates are also higher with cilta-cel than any of the other agents, and it induces MRD negativity at higher rates as well. So you seem to get better responses, deeper responses. And then there's been updated data since the initial trial was published with cilta-cel, and it seems like the median duration of response. So these are patients who do respond. They respond for over 20 months, and even at 24 or 27 months, half of patients had still not progressed. So that's significantly better than either ide-cel or teclistamab, and certainly better than belantamab as far as we can tell. And that seems to be the biggest difference. And why that difference exists is something that we still don't fully understand, and there's a lot of work going on trying to understand that difference and how we can make it better. So these agents do have some real potential side effects. So the first is what we call cytokine release syndrome, So when the T cells are infused into a patient, they start to grow, develop, and target the multiple myeloma. And in so doing, as they're growing, they release what are called cytokines. These are inflammatory molecules that help trigger the immune system. And these are some of the same things that are released when you have a cold or a flu, and they're probably what cause some of those symptoms. So they can cause fever, body aches, and a lot of those symptoms that come with a flu. And that's really the earliest signs of cytokine release syndrome, and probably the most common thing. So most patients do develop a fever and will have what we call constitutional symptoms, so body aches, fatigue, chills, things like that. But if the cytokine release syndrome is more severe, it can produce more severe symptoms. So that includes low blood pressure, difficulty breathing, and even shock or organ damage at very severe levels that can require very strong medications, as well as transfer to the intensive care unit. Now, we've gotten better at identifying cytokine release syndrome, and because it seems to be mediated by these cytokines, we've also identified ways that we can target it and sort of cut it short. And so the way we do that is we have anti-cytokine drugs. So these are drugs like tocilizumab or siltuximab or anakinra that can target the cytokines. And then we can also sort of stop the T cells from dividing and growing quite so fast. And to do that, we use therapies that target the T cells, like dexamethasone. One other complication of the CAR T cell growth is what we call neurotoxicity, or the immune effector cell associated neurotoxicity syndrome, ICANS. And this is often associated with the cytokine release syndrome, but can also be separate, and seems to be driven by processes we don't fully understand, but maybe inflammation that leads to damage of the small blood vessels within the brain. That can lead to swelling of the brain as fluid enters it, and can lead to confusion, seizures, and other complications. Now, this is more common with the CAR T cells used in lymphoma and leukemia than it is in multiple myeloma, but it is something that we still do see, and it's treated largely the same way, because we do think that it is mostly mediated by these inflammatory cytokines. So we use the same kinds of treatments like tocilizumab and dexamethasone. And in most cases, it's quite manageable and recovers to normal fairly quickly. So here's just a summary comparison of ide-cel, cilta-cel, and teclistamab. Because teclistamab also targets T cells, it is able of causing some of the same symptoms, although it does seem to do so at a lower frequency. So the most common side effect for all of these agents is actually low blood counts, but that can be managed fairly easily, and that's seen in most patients. And then CRS, as you can see, is quite common, between 80 and 90% with the CAR T cells, a little bit lower, around 70% with teclistamab. It occurs at slightly different timings depending on the different drugs, so about 1 day after infusion for ide-cel, and about a week after infusion for cilta-cel, whereas for teclistamab it's about 2 days after treatment. And then neurotoxicity of any kind can occur in about 20% of patients with CAR T cells. It's a little bit lower with teclistamab, and again, similar onset to the CRS. More severe neurotoxicity that we really worry about actually is quite rare with all of these agents. There has been some concern about a delayed neurotoxicity, which was seen in a few cases with cilta-cel, and that's something we don't totally understand, but that we're working on trying to figure out ways to prevent. So I'm going to talk a little bit more about future directions. I think most I'm going to talk about other targets for CAR T cells, but there's a lot of other approaches that are being developed, including new CARs that target multiple antigens, that use other types of immune cells, rapid manufacturing approaches so we can get these cells to patients faster, and things like allogeneic CARs, where we can make CAR T cells that are sort of off the shelf. And then finally, of course, combining CAR T cells with other therapies to see if we can make everything more effective. So one of the other targets that has recently been discovered is called GPRC5D, and this was discovered by looking for proteins that are expressed on multiple myeloma cells but aren't really expressed on any other cells. And it was sort of one of the top hits, and here's data. It's really expressed on multiple myeloma cells and then on some skin cells, and that's about it. And so that led to CAR T cells that target GPRC5D, and this has been tested in a phase 1 clinical trial now. And again, the outline for this clinical trial is very similar to the others. It was tested at 4 different cell doses, and again, patients had their cells collected, manufactured, and then received standard lymphodepleting chemotherapy. And the early data would suggest that this is a very promising target. So here are responses, and you can see that the majority of patients had responses. Some patients have had long-term responses that are still ongoing, particularly at the higher doses of infusion. And you can see that this is quite impressive. This is a PET scan from the first patient that was treated. You can see these very large masses that are very, very bright on the PET scan, and they have completely resolved following treatment. And so overall, it's still early. They've only treated, I think, less than 20 patients, but the response rate was around 70%, and about a third of patients had a complete remission. And the toxicity is largely what we would expect for our CAR T cells. So most patients had the cytokine release syndrome, although very few had severe cytokine release. And the neurotoxicity is a little bit different. There wasn't very much of the standard neurotoxicity, but there were 2 patients that developed what's called cerebellar toxicity, which causes dizziness and sort of feeling of vertigo and unsteadiness. And it's, again, still not totally clear how common this will be and exactly what causes this. But I think this is a very promising target, and the clinical trials are ongoing, and hopefully it will be available more broadly in the near future. So with that, I'm just going to conclude. So I think CAR T cells are a very exciting new therapy for patients with multiple myeloma. They're now available in the clinic, and so we're using them more and more. But I think there are many challenges ahead. I think for many patients, the manufacturing time is a real challenge. It's very difficult to keep the multiple myeloma under control during the time it takes to manufacture the cells. And for many patients, they relapse after a year or 2 years, which suggests that there's resistance to the treatment that we have to overcome. And so figuring out how to overcome that resistance is one of the big challenges going forward. And so part of that will be combining CAR T cells, either combining 2 different CAR T cells to target different antigens, combining a CAR T cell and a bispecific, combining CAR T cells with standard multiple myeloma therapies to improve their activity. But I think we're still figuring that out. I think the other big question is whether or not they will work better if we move them earlier in treatment. So could we replace stem cell transplant with CAR T cells and have them work better? I think that's still a big question that needs answering. And then if we can get them to work better, can we finally cure multiple myeloma? And I think we're all very cautiously optimistic.